In this video, we're going to talk about a bit more detail involving how neurotransmitters relate to drugs and how those drugs affect receptors that neurotransmitters naturally interact with. So in the previous videos in this playlist, we've gone through resting membrane potentials, talked about how action potentials are generated, and gotten into some details about exactly what's happening at a synapse like this one. The next question is, how can we use drugs and pharmaceuticals to mimic neurotransmitter effects? Right? And so the first thing to point out is, in order to have an effect on a neuron, you have to both release the neurotransmitter, and that neurotransmitter has to bind a receptor. The receptor has to lead to the opening of an ion channel, right, that lets some sort of ion either flow in or out of the cell. And those responses can either be excitatory or inhibitory. So thing one is to remember, when we use the term excitatory and inhibitory, we're talking about neurotransmitters. But in reality, whether the neurotransmitter is excitatory or inhibitory really depends on the receptor, right? Because if this receptor happens to be a sodium channel, well, it's excitatory, right? Because sodium has a positive charge, and those positive charges are entering the cell. If this channel happened to be a potassium receptor that let positively charged potassium leave, that would be an inhibitory neurotransmitter. All right, and so the way that it makes, I think, the most sense to remember this is the receptor determines the response. And you don't know which ion channel is going to be open in which neuron. And so when I say, oh, we're releasing dopamine in the brain, well, in some areas, dopamine opens sodium channels and it's excitatory. And in other areas, dopamine opens potassium channels and it's inhibitory. So in this picture, this coiled green structure is dopamine receptor, one of them. And you can imagine that receptor being inserted into the cell membrane of a neuron. This little colored structure right here, that is actual dopamine itself, right? And this is looking at it from a side view. So you would expect, if this is a sodium channel and these represent sodium ions, you would expect them to diffuse this way into the cell. This is like an aerial view, a top-down view. And if you look very closely, there's a little pore in the center. That's the opening where you expect sodium ions to diffuse through the pore, through the channel. Okay. This receptor is what gives the neurotransmitter specificity, right? So the receptor is what our drugs are going to target, right? So when you make or take a pharmaceutical, it can act in one of two ways. Some pharmaceuticals act as agonists and some as antagonists. And all those terms mean is that an antagonist is exactly what it sounds like. It somehow blocks the effect of the neurotransmitter, whereas an agonist mimics the neurotransmitter and makes even more of that response happen. And so if you had a drug that was an agonist to dopamine, it means that that drug would be able to bind this receptor and open the channel in the exact same way that dopamine does, right? If it was an antagonist, you could imagine it binding the channel, but not actually opening it and preventing real dopamine from getting the job done, right? So we can put all this together by imagining a few different possibilities. On the neurotransmitter side, we have two different options, right? Neurotransmitters can either be excitatory or they can be inhibitory, and those are your only real major options. On the drug side of things, and this doesn't really matter whether they're illegal drugs or pharmaceutical drugs, it's the same principle. They can either be agonists or antagonists, right? And that gives us four possibilities. If you had a drug that is an antagonist or an agonist to an excitatory or an inhibitory neurotransmitter, it's going to produce very different results. So agonist to an excitatory neurotransmitter, that's this category. Agonists mimic. So that just means that we're going to get more excitation. If you had an agonist to an inhibitory neurotransmitter, that means that you're just going to get more inhibition, right? An antagonist to an excitatory neurotransmitter means you're blocking excitation. The end result is going to be inhibition. And the really confusing one, if you antagonize an inhibitory neurotransmitter, you're blocking inhibition and you get a double negative and it winds up being excitatory, right? So the idea here is your drug is one of these two things, but the neurotransmitter that it targets is one of these two things. That's how you get your 4 possible combinations. All right, so as an example of this, these agonists to excitatory drugs, opioids, fit this category. They're excellent pain relievers because they stimulate neurotransmitters that cause pain relief throughout the brain. On the flip side, Narcan, that's used as an antidote of sorts, is an antagonist to that exact same receptor. So they fight each other. If someone has too much agonist in their system in the form of the opioid, we can give the antagonist, the Narcan, to counteract it, right, and reduce its effect. Okay, the next general principle to think about here is a concept called affinity. Different drugs can bind the same receptor, but have very different affinities. And you can think of affinity as meaning binding strength, right? So an example of this is if you imagine 2 different opioid drugs like OxyContin and fentanyl as an example. If you give someone a few hundred milligrams of OxyContin, it'll cause effective pain relief and they'll be fine as long as they don't abuse the drug. If you gave someone a few hundred milligrams of fentanyl, it would be a massive, massive overdose, even though those 2 drugs bind the exact same receptor. The reason behind that is fentanyl has a much, much higher binding affinity. It sticks to the receptor much more tightly. And the reason for that is how well these 2 things, the molecule and the receptor, stick to each other are determined by shape and charge. And we want them to be opposite. So think of it like puzzle pieces that are magnetic, right? The charge is the magnetic part. The shape is like the puzzle piece part. When a drug or a neurotransmitter has a very, very high affinity, it just means that their shapes and charges line up so perfectly opposite of each other that they stick really tightly and small amounts of drug can go a really, really long way. So that's why you can't standardize the use of all those drugs with the same dose because they have different binding strength and different doses will lead to very different responses. Right. The other thing that this can lead to is something called tolerance. Everyone knows the concept of tolerance, right? It's where you take a drug and over time that drug starts to have less and less of an effect. And so the person will have to increase the dose if they, want to keep getting that response. And in principle, what's happening on a molecular level is if you're stimulating, right, pretend that my red dots here are an agonist that mimics this neurotransmitter. If it binds and opens these channels, you're going to get a lot of stimulation of this postsynaptic cell. Now remember, these receptors are proteins, and proteins are encoded by genes. So there has to be a gene in this cell that codes for that protein. And the cell can decide how much it wants to turn the activity of that gene on or off. And so when a person takes a drug over and over and over again and hyperstimulates those receptors, what do you think this cell does? It says, wow, I'm being wildly overstimulated. I probably have too many receptors. And so just like a person who's being yelled at, plugging their ears, the equivalent for the cell is removing some of these receptors. And now that there are less receptors, there are less of them that can bind the neurotransmitter. And so what does the person have to do if they want to keep getting their high or their pain relief or whatever they're looking for? They have to take more and more drug to hyperstimulate the receptors that are left. And then what does the cell do? It takes away even more, and the dose keeps going up, right? And then when this person finally tries to get off the drug and go through detox, the pathway is going to be so incredibly depressed because now not only have we removed a lot of receptors, right, we don't have the drug anymore. And so the naturally occurring neurotransmitter is going to have a very, very hard time getting the job done. That's why opioid addicts that try to stop taking their drug, everything feels painful because their internal pain relief system is now incredibly weakened after having all these receptors removed. Okay, so From here, we're going to do a quick summary of a few different common neurotransmitters. It's by far, not anywhere near an exhaustive list, but some examples to give you an idea of how neurotransmitters operate. All right, so neurotransmitters fit a lot of different categories, but by far the most common category that you're going to see are these monoamines, and they get their name because they're synthesized from single amino acids that act as precursors. And those amino acids are just modified. So monoamines, dopamine, norepinephrine, epinephrine, and serotonin are responsible for a pretty large proportion of the neurotransmitters released in the CNS. And what's interesting is they're not as much regulated by how much is released as a synapse as they are by reuptake. And so since these neurotransmitters are released at relatively consistent levels a lot of the time, instead of controlling how much leaves the axon terminal, we control their reuptake and degradation, right, through enzymes, right, like monoamine oxidase and this catechol-methyltransferase. These are enzymes that degrade the neurotransmitter when it needs to be gotten rid of, right, as opposed to signaling the release when it needs to enter. Okay. So they all share a fairly similar chemical formula. And the point of showing you this picture is just to point out that very, very small changes in chemistry can lead to very big changes in how they bind the receptor. Okay, so as an example, I like dopamine for a reason that I mentioned earlier. Dopamine actually has 5 different receptors, and some of them are excitatory and some are inhibitory. The point this proves is that the same neurotransmitter can have very different effects in different parts of the brain. And the reason this is possible is that some of those dopamine receptors are sodium channels and other ones are potassium channels, right? The ones that are sodium channels are excitatory and the ones that are potassium channels are inhibitory. And so if you give a drug that impacts dopamine in some way, you can't really say that you're having an excitatory or an inhibitory effect on the entire brain. You're actually having a mix of both effects in different brain regions. And that's why the effects of those drugs can be incredibly variable and complex because you're affecting so many different brain regions in different ways. All right. In general, dopamine is associated with reward and motivation. And a lot of times these are given as the 4 Fs, feeding, fighting, fleeing, and for the sake of keeping this PG-13, fornicating. So anything that drives that reward motivation and can act like an addiction, when you get the response that you're looking for, whether it's finding food or gambling or whatever the person is seeking, parts of your brain get a little spritz of dopamine that's effectively a reward to say, yep, that feels good, I like it, let's do that again. And that can lead to addictive behaviors because the more a person gets that stimulatory feedback from dopamine, the more they want even more of it. All right, so that makes it a potential target for treatment of depression, to try to give people a low-level, steady stream of dopamine, to try to raise their mood across the board, and avoid those big, strong bursts being the only way that they're getting a hold of it. All right, serotonin is also used as a depression treatment. We mentioned earlier SSRIs, selective serotonin reuptake inhibitors. Those are drugs that prevent the recycling and removal of serotonin from a synapse to keep it trapped in the synapse longer and keep having an effect. Right? And since serotonin very broadly regulates a lot of things like sleep and mood and pain modulation, right, it makes a good target for depression. So this isn't like dopamine where it's giving you that sudden burst of pleasure feeling when you find the thing that you've been looking for, like from a behavioral seeking standpoint, but it's regulating a lot of sort of background physiological variables like mood and sleep and memory that can still play a role in the depression process. Okay, a few others just to throw in there. Gaba is by far the most common inhibitory neurotransmitter in the brain. It's a really interesting example of agonism. Alcohol increases activity of GABA, and so it acts indirectly like an agonist, and that makes sense, right? Because when someone has too much alcohol in their system, almost everything that they do slows down. Motor function, cognition, speech, it's all slower because we're increasing the activity of an inhibitory neurotransmitter, making it more and more difficult for their synapses to reach threshold. All right. And then lastly, I wanted to mention endorphins. These are sometimes called endogenous opiates. So opioid drugs are mimicking endorphins in a lot of ways and activating the receptors. And this makes sense because endorphins inhibit pain. And so we use opioid drugs to further inhibit that pain response. But that's also why they have a high potential for addiction, is because they make people feel so good and effectively impervious to pain. And so morphine, heroin, codeine, methadone, fentanyl, all the way down to opium, all of those drugs are agonists to this endorphin category. All right. I hope this is helpful. This is pretty much the end of my series in action potentials and synapses and neurotransmitters. And in the next video, we'll be moving on to brain anatomy. So make sure you follow along the playlist and subscribe so you can watch all the videos in the correct order. And I'll see you next time to start going over introduction to anatomy of the brain.